Hi, and welcome to the GSDSEF video on how to complete ISEF Form 3 Risk Assessment presented by the Student Leadership Board.
In this video, we'll be going over how to complete ISEF Form 3 Risk Assessment, and specifically how to answer questions on Form 3, and any additional forms which may be necessary.
Let's start by going to Table 4B in the gsdsef.org website and clicking on How to Complete ISEF Form 3 Risk Assessment.
So let's start by reading through Section 1 of how to complete the risk assessment form. So in any general project which involves hazardous materials, activities, or devices must include Form 3. Some examples include dangerous activities, chemicals, model rockets, lasers, radiation, etc. And please note that consumable alcohol distillation, cigarettes, loading/reloading ammunition is prohibited in all GSDSEF projects. Also, if there are any biohazards, you will need to fill out Form 3, such as fungus, bacteria, virus, etc., anything that makes you sick. And if you are using bacteria, it must be grown in school or research lab. If you're collecting any samples from the environment, it may be collected at home, but not grown on agar plants at home. DNA from an organism, anything coming from a human or animal body, and no organism may be grown on blood agar or chocolate plates.
Now we are going to the ISEF guidelines. If you need more information, read ISEF guidelines regarding specific hazards information on pages 15 to 21. So we're just scrolling to pages 15 and 21, and just make sure to read through these on your own time.
Finally, talk to your teacher or supervisor about potential hazards in your project and decide which hazards are present. It is especially important that you discuss any hazards with your teacher or supervisor, since they will also be signing the form.
After you have determined with your supervisor that your project has hazardous materials or living things, then go to Table 4B on the gsdsef.org website and click on How to Complete the Risk Assessment Form. Read numbers 3 to 6.
So you can go through these on your own time, and there will be a downloadable Form 3 directions, which will include directions to fill in the questions. So we go to ISEF Form 3 Risk Assessment.
So here is Risk Assessment Form 3, and from here we're going to go through each section of the form and describe what must be included in depth.
So first, let's go through the student names and title of projects.
Note that ISEF forms are fillable PDFs. That means you can type in the areas after directions. If you have a team partner, make sure to fill in both names.
So in this project, we'll be assuming Albert Einstein and Nikola Tesla are entering a project in the GSDSEF. So in bold, it says to be completed by the student researcher in collaboration with designated supervisor/qualified scientist. So what this means is that you will be completing this form, but make sure that you review it with your designated supervisor or qualified scientist.
So now let's go through question 1. Here you will identify and assess the risks and/or hazards in your project.
Now let's take a look at question 2. For 2A, give a detailed list of hazardous chemicals or devices and a detailed description of the activities you are doing. This is your procedure.
And for 2B, give the scientific name of the microorganisms you are using, unless they are exempt from pre-approval.
So now, see the rules on potentially hazardous biological agents, page 17 of ISEF guidelines. I have it pulled up here. Alright, so back to the form.
Let's move on to question 3. After assessing the risks in your project, describe the safety precautions you will take to reduce the risks.
For chemicals, it would be a good idea to look up the MSDS, or Material Safety Data Sheet. These are available online from various chemical supply companies. Consult at least 2 before describing your precautions with chemicals. Alright, so we can look at the Flint Scientific and Sigma, and they have an MSDS where you can find your data sheet. So you can go through this and check which chemicals may be dangerous or exempt.
Moving on to question 4. So your description of disposal procedures for bacteria or toxic chemicals must be detailed. Again, a good source for the proper disposal of chemicals would be the MSDS, which we just showed. Bacteria must be autoclaved or sterilized for a minimum of 60 minutes at 15 psi and a temperature of 121 degrees Celsius. Alternatively, bacterial cultures can be placed in a 10% bleach solution in plastic bags and double sealed. Bacterial cultures from either disposal method can be placed in regular trash.
Alright, so if you have used MSDSs or any other source material such as journal articles to describe your procedure, safety precautions, or disposal procedures, list them here. They must include the name of the source and the URL if you got them from the internet.
After you have completed the form with the help of your teacher, supervisor, or qualified scientist, have them fill out and sign the box at the bottom of ISEF Form 3.
Now, let's see if you need any other forms. If your project involves no biological hazards, you will only use Form 3.
However, if you have used any biological hazards, you must fill out either Form 6A or 6B depending on the nature of your project. And once again, this is in addition to Form 3. So Form 6A is the potentially hazardous biological agents form, and Form 6B is the human and vertebrate tissue form.
Also, if your project involves human participants who might be mentally or physically stressed by the project, then Form 3 is necessary along with Form 4, human participants. Thank you for watching. If you have any questions, please contact us using the Contact Us Online button at the bottom of any gsdscf.org webpage.
Thank you for tuning in.